Okay, so I'm John Scahill. I'm Biological Secretary of the Society. I have to ask you to make sure your mobile phones are switched off and to tell you that there are no planned fire evacuations, but if there is a fire, you leave through that door. Okay. And of course, you'll have gathered from this thing that the event is being webcast and recorded. Okay. So this evening's lecture, the Croonian Lecture, is the Royal Society's premier annual award in the biological sciences. It was planned by William Croon, one of the original fellows of the Society, and was awarded first in 1738. It's my pleasure to welcome this year's lecturer, Brigid Hogan. Brigid is very well known for her molecular and genetic research in developmental biology. Through it, she's made numerous observations that have greatly increased our understanding of how specific cells are generated, how they assume their shape, and how they develop into organs. Her research on the lung and the cells from which lungs are formed is highly relevant to considerations of the treatment of diseases of these important organs. Brigid is the George Barth Geller Professor in the Department of Cell Biology at Duke University Medical Center in Durham, North Carolina. She did her PhD in Cambridge and her first postdoctoral research at MIT. Following that, she led research programs at the Imperial Cancer Research Fund and at the National Institute for Medical Research in Mill Hill, where she was head of the Laboratory of Molecular Embryology. From there, she moved to Vanderbilt Medical School and to her current position at Duke in 2002. Brigid is past president of the American Society for Developmental Biology and the American Society of Cell Biology. She's a fellow of the Royal Society and the American Academy of Arts and Sciences, and she's a member of the National Academy of Sciences of the United States of America and of the Institute of Medicine. Her lecture this evening is entitled, How Embryos Build Organs to Last a Lifetime. Ladies and gentlemen, Brigid Hogan.
Well, thank you very much indeed for that introduction and for the very great honor to speak tonight about the ongoing and past work of my lab. Now, this famous picture of Botticelli, I think, beautifully shows the joy that the zephyrs have in blowing flowers through the spring air. And this ability to blow flowers depends upon the structure and function of their lungs. And the ongoing ability to do this depends on its past history of this organ, as well as its future maintenance. And the past history is the embryonic development of this really complex organ with a very few founder population of cells in the embryo. And as David Horowitz pointed out to me, that actually my title was a little bit wrong because these organs don't entirely last a lifetime going forward. They do run out eventually with age, but nevertheless, we're beginning to understand how stem cells resident in these organs can at least hold back the ravages of time and cell loss due to disease and things like infection in the lung. Now, these considerations aren't unique to the lung. And in fact, they apply to many of the organs in the body, and in particular, those organs which are constructed, like the lung, from a system of branched epithelial tubes closely aligned with blood vessels and with stroma. And what we learn about the lung can be applied to other organs like the kidney, and what we learn from, say, the breast can also be applied to understanding the lung. So my talk is divided into two parts. First of all, I'm going to talk about how lungs develop in the embryo and the process of branching morphogenesis as a kind of basic principle of organogenesis. And then the second part, I'm going to talk about how lungs maintain themselves using resident stem cells and the microenvironment of these stem cells. But first, I need to just remind you about the structure of the lung because of the relevance that that's going to be to the embryonic development. And so just very briefly, I said that it's made up of a system of branched tubes. And the most important one, of course, is the epithelial tube, which derives from the endoderm. And it's a hollow tube, and it branches, giving finer and finer little bronchioles. But right down then in the periphery, it bursts out into millions of tiny air sacs where the gas exchange takes place. Now, although this epithelium is continuous, the kind of structure of the cells making up the tubes are rather different in different regions. In the places where the air is being conducted, then we have it lined by what's called a mucociliary epithelium. And about half of the luminal cells are secretory cells, which are secreting a sort of mucus which traps bacteria and particles. And the other half of the cells are these really beautiful multiciliated cells, which are all beating together towards the mouth and removing the mucus. Now, this kind of arrangement of cells wouldn't be very good if it was down here in the gas exchange region, because the oxygen has got to diffuse across the cell layer. So down here, we have a rather different arrangement of epithelial cells. It's more like a sort of honeycomb of these millions of tiny sacs made up of two different kinds of epithelial cells, the type 2 cells, which make surfactant protein, which lines the alveoli, and then these very flat type 1 cells, which are designed for allowing gas exchange. Now, I said that there was an epithelial component. There's also mesodermal populations in the lung, which are very important for its function. And the most important of these are the blood vessels. And these are closely aligned to the tiny branches. And in fact, they grow and branch alongside the epithelium. It isn't like you don't construct the epithelium first and then plumb in the blood vessels later. They develop alongside each other. And then the capillaries have to come into very close contact with these air sacs. And as we'll see, this is one of the most challenging parts of the organogenesis program, is to get these blood vessels into close proximity with the epithelial cells. And then just very briefly, there are other mesodermal populations. They give rise to the cartilage rings, which keep the airs open. And then smooth muscle, which is tightly around the bronchioles, and constriction of this can reduce airflow under some circumstances. There's also an interesting layer of cells called the mesothelium around the outside that stops the lung from sticking to the thoracic cavity. And then also there are other cell types which I don't have time to talk about. It's extensively branched innervation, and there are lots of macrophages, for example, which are inside these alveoli, keeping them clean. So this is a very complex organ. It's got these branched epithelial tubes, it's got the blood vessels, it's got the mesoderm. How does this develop? And this is one of the things which fascinated me most about studying the lung, was this remarkable fact that actually this complex organ just arises from two small buds. Really amazing, epithelium surrounded by a simple layer of mesoderm. And these buds appear quite early in development, about 5 weeks in the human and 9 days in the mouse. And one of the challenges is to understand how the embryo knows where these buds should form in the right place and the right number. I don't have time to go into this completely, but just to schematize it here, what we know is that there are growth factors and signaling factors and transcription factors that together set up coordinates in this early foregut tube. This, in the embryo at this time, the epithelium is a simple single tube surrounded by very undifferentiated mesoderm. There is patterning within this tube such that there are gradients of morphogen setting up an anterior-posterior axis, a dorsal-ventral or back-to-front axis, and a right-left axis. And the cells in this tube are able to read the instructions from these coordinates. And this is where the lung bud will then form, where the transcription factor NKX 2.1 is switched on in the ventral part of this foregut. And this then forms these two primordial buds, which start to grow out and start to branch in a very stereotypic manner. Now, there's another very interesting morphogenetic process that's going on here that I'm not going to just very briefly allude to, which is, I said that this is a single tube, but we all know that there are two tubes, one, the trachea leading into the lungs, and another one, the esophagus leading into the stomach. And so this tube has to split into two, and there's another very interesting morphogenetic process which produces this effect. And my ex-postdoc, Jianwen Cui, who's now got his own lab in the University of Rochester, was one among others who showed that this splitting process is genetically controlled, and there are genes like SOX2 and noggin, in which, in the mutations, in mice which have mutations in these genes, there's a failure of this separation. And then this results in a fairly common birth defect called tracheoesophageal fistula, where there's small junctions between the trachea and the esophagus. Anyway, we've got these little buds growing out, and this then continues to branch by a process known as branching morphogenesis. This has been studied first. Ross Metzger in Mark Krasnau's lab was the first to show that these early branching is actually quite stereotypic. Every lung dissected from an embryo, a mouse embryo, will have exactly the same pattern of primary and secondary buds and branches. More recently, this has been viewed in much more detail than he did, using the technique of optical projection tomography, or OPT, which is really a very powerful tool and actually was first developed by James Sharp in the MRC in Edinburgh. And these images were actually given to me by Ji-Chao Chen from the MD Anderson Cancer Center in Houston. And these are mouse lungs that he had stained with an antibody against the transcription factor SOX9, which is just expressed in the cells in the growing tips. And then E-cadherin, which is marking the epithelial tubes as they branch. SOX9 is also in the developing cartilages here. So it was thought that and shown that the early branching is stereotypic. What about this later branching? Well, with optical projection tomography, it's been possible to actually view this in more detail. And this is not from my lab, but I really wanted to show it because I think it's so beautiful. Ian Smith in Monash University developed a way of digitizing these branches. And this is a movie taken from his paper from that great journal Development, in which it's homing in on a particular region of one of the lung lobes. And here he has shown this skeletalization, as he's calling it. And they've used this program called the Tree Surveyor branch analysis program to sort of digitize this and then able to compare one lung with another. And using this, they've shown that although the early branches are stereotypic, then the later ones are actually more space-filling. And as I said, what we can learn from lung can also apply to some other organ systems. And Ian Smith has applied this tree surveyor program to look at the branching of the kidney, which, like the lung, forms from a little bud branching into surrounding mesoderm, and then it grows and becomes more complicated. But by applying this tree surveyor program, he and Melissa Little have able to show that actually they can describe the total branching program of the lung through several days of development, and showed that actually it's much more stereotypic than expected, although there is a certain amount of remodeling that's going on. So this is all very descriptive. What about the actual molecules and signaling factors which are involved in controlling this branching and allowing the outgrowth and the interaction between the mesoderm and the epithelium so that the development of each is coordinated? And a lot of the early cues for this came from studies in my lab and others, and here are the postdocs who were involved in this, and Molly Weaver, a student, looking at the expression of specific families of transcription factors and signaling factors in the developing lung by a method that enables you to see the messenger RNA for some of these genes. And what became clear was that there was a reciprocal signaling between the epithelium and mesenchyme. For example, the gene encoding sonic hedgehog, a secreted signaling factor, is expressed in the epithelium, whereas the gene for patched, one of its receptors, is expressed in the mesoderm surrounding the little bud, suggesting that hedgehog is signaling to the mesoderm, and indeed, in mutants that don't have sonic hedgehog, the lungs fail to develop properly. And one of the most interesting patterns that we saw, and I remember the excitement we had when Saverio Belushi first did this in situ hybridization for the gene encoding FGF10. It's a well-known family of signaling factors. FGF10 is expressed in the mesoderm. It just localized above one of these budding and branching epithelial tubes. And this expression of FGF10 continues in the lung throughout the branching process, and often the FGF10 will be seen to be over where a new bud is going to form. Now, FGF, when it signals to the epithelium, will trigger a downstream cascade of signaling factors. One of these is Sprouty2, and you can see from this in situ hybridization that Sprouty2 is in the epithelium, which is what you'd expect if FGF was signaling from the mesenchyme to the epithelium. Since these early studies, hundreds of genes have been shown to be expressed in organs like the lung, and if any of you are interested, there's a very beautiful, interesting website with many of these patterns. However, we were challenged to say, well, okay, these patterns are very beautiful. What do they mean? Is there functional significance in FGF being expressed in the mesoderm? And some of the early experiments were done by Molly Weaver in my lab. This is just describing one of the ones that she did, which was to cut off the tips of these early embryonic lungs and separate them into the endoderm and the mesoderm, and to put the endoderm into a little gel, a matrix gel known as matrigel, in which the epithelium can grow if it's given the right combination of growth factors. Without an FGF, they won't grow, but with FGF, they do grow. And what Molly did was to put FGF on a bead, a localized bead here, a heparin bead, and she found when she did this, the buds would grow and would move towards the bead and even try and sort of engulf it. Yuru Liu also looked at some of these other downstream signaling cascade components, and phospho-ERK, which is well known to be part of the FGF signaling, is just expressed. This green is showing the epithelial bud, and you can see these most tip cells are expressing phospho-ERK. And then studies, a sort of gold standard for whether a gene has a function, is what happens if you knock it out or if you make a null mutation. That was not done in my lab, but in other labs, they knocked out FGF10 and the receptor for FGF10 in the epithelium. And in both cases, the mouse embryos were born without any lungs. Now, I don't want to make this seem too simple, this idea, because actually there are other players in this branching to controlling the outgrowth of the buds. David Ornitz's lab has shown that FGF9 made by that outer mesothelial layer is actually important in maintaining the ability of the mesoderm to make FGF10. So FGF9 is an important player. And I tell you, there's still a great deal more to learn. So people who look later on the web will be directed towards nice papers which have been showing how the FGF signaling control the outgrowth of one of these buds by orienting cell divisions and so on. But now we have this branching process going on. How do we coordinate this branching morphogenesis with the differentiation of the epithelium? Because I told you in the more proximal, what we call the proximal epithelium, you've got this mucociliary epithelium, and then in the distal part, we've got these very thin honeycombed air sacs. How is this coordinated with the branching process? And the first ideas for this actually came from the work of Emma Rawlings, who's in the audience here, and who's now got her lab in the Gurdon Institute. And this is a picture that she gave me of a section of a mid-gestation mouse embryo in which this is revealing the expression of SOX9, you remember, is in these little tips. But further back, we have the expression of this green cells of FOXJ1 GFP. Now, FOXJ1 is a transcription factor that's only expressed in developing ciliated cells. And in this transgenic mouse we made, it's, the promoter for FOXJ1 is driving the expression of green fluorescent protein. So everywhere where a cell is going to start differentiating to a ciliate cell is going to be green. And I'm going to come back to this transgenic line a little bit later. So Emma's idea was that these tip cells, which she had showed actually express transcription factor ID2 with the same sort of pattern as SOX9, these are undifferentiated proliferative progenitor cells. And then as these cells move away, the daughter cells move away from the tips, they start to differentiate and will switch on FOXJ1. They also start to express another transcription factor, SOX2, shown here. And then these cells will start to differentiate into those secretory and ciliated cells. And work from other labs has been shown that the patterning of these and choices of the ciliated and secretory cells can be controlled by an important role being played by notch signaling here. Then the question is, though, that this is going on during the first stages of development. These are differentiating into the ciliated and secretory cells. As development proceeds, these tip cells sort of change the fate, and the daughter cells now give rise to these honeycombed alveolar cells. And so the fate of these cells has to switch sometime from making multiciliated and secretory cells to making type 2 and type 1 cells. And I'm not going to go into a full story about this, but again, I would direct you to a very recent paper from Ji-Chao Chen's lab, which is proposing that signals, glucocorticoid signaling from the mesoderm might be playing some role in controlling the fate of these progenitor cells at the tips. So that brings me now to the very last slide for this first part about the development. And this is really the most challenging stage of the embryonic development of the lung, and the one which is really highly relevant to the care of premature babies. Because what we have to do now in the last stages of lung development is to take these tips, which still have a few SOX9 and ID2 positive cells at the tips, and we've got to convert them into these mass of little air sacs containing type 2 cells making surfactant and these very thin type 1 cells. And this involves a process which is a slightly different morphogenetic process than budding. And as I said, one of the key things is to get the blood vessels in there in close contact with these thin type 1 cells. So premature babies now are born actually at this stage of development, and their lungs have to go through this very exquisite process of making alveoli under conditions in which they're being artificially ventilated and often will have infections and so on. So it's very important to understand what is going on here, and we're only beginning to get at this at a molecular level. So what has to happen is that the blood vessels go in, and they, at the same time, populations of mesodermal cells, fibroblast cells, are also moving in, and they're doing two things. One is laying down that elastin network, which is going to be very important for the elasticity of the lungs. And another thing, as I'm going to explain, is they're going to be producing a little niche for the stem cells, which are in this alveolar region. Now we're beginning to understand what's going on here, and I just want to highlight a couple of things. One is that these SOX9 cells and ID2 positive cells, which are round at the tips, don't seem to persist beyond the first few days after birth. It isn't as though this alveolar region contains a population of undifferentiated cells which can act as stem cells. As far as we know, they sort of hand over this role of being progenitor and stem cells to the type 2 cells. These cells which are actually making surfactant protein and expressing surfactant protein genes. The other thing is, though, that the SOX2 expressing cells do persist, and they continue to express this gene in the airways and actually in the larger airways as well, which are not shown here. And we are just beginning to investigate also this other transcription factor, HOPX, which is a marker for the type 1 cells in the alveolar region, and together with John Epstein's lab in Philadelphia, we are beginning to understand how this HOPX gene is switched on early during this process of what's called alveologenesis. So as I said, this is a very key part of organogenesis, and I will again refer you to some other recent papers as well, looking at the molecular processes in this differentiation. So now to switch to the second part of the talk, which I may have to hurry up about, and that is how are lungs maintained at least as long as possible during our lifetime, and how do they repair injuries which might be caused by wear and tear or by infections or by exposure to environmental toxins and so on. So this one take-home message for you is going to be that there is no one kind of stem cell in the lung. There are stem cells in different regions of the lung which differ in their properties. And because some of them are a little bit different from the sort of stem cells that one thinks about in the crypts of the intestine or in the skin, I just want to say that we define a stem cell as a cell or a population of cells. I'm not going to go into whether there's heterogeneity, but a population of cells that can self-renew, can make more of themselves over a long period of time, but can also, under certain circumstances, differentiate into another kind of cell and give rise to a more specialized cell of a different type. And so if we take that as a definition of a stem cell, then there are different kinds of stem cell in the lung. Up here in the mucociliary epithelium, where there are these multiciliated cells and secretory cells, in the mouse trachea and in the human lung throughout the airways, which are the same diameter as this mouse trachea, then there is this population of basal cells, which express many of the same kind of genes as in basal cells in other organ systems like the mammary gland and the prostate and the skin. They express a transcription factor P63, keratin 5, and nerve growth factor receptor on their surface, which is, and these have been shown by us and others to be able to self-renew over a long period of time and to give rise to ciliated and secretory cells. But down in the smaller airways, there are no basal cells in the normal mouse lung. There are instead mostly cells, ciliated cells, or these secretory cells, which are now called club cells, which express genes for some of these sort of secreted glycoproteins. And Emma, when she was in the lab, has shown that these can self-renew over a long period of time, but can also, under certain conditions, give rise to ciliated cells. So they meet the definition of a stem cell, even though they're not totally undifferentiated. And then we, as I said, in the alveolar region, there aren't any of these SOX9 progenitors, or ID2 progenitors. Instead, there are these type 2 cells which express genes for surfactant proteins, and we have evidence that these can self-renew over a long period of time and can give rise to type 1 cells. So that's the general picture. However, because turnover in the lung is rather slow, people use models in which they will mimic certain kinds of conditions in the human lung where cell types are destroyed, and then say which cells will respond by making replacing the lost cells. And what has been found is that there's quite a bit more plasticity than was previously thought. And J. Rajagopal in Harvard recently showed that if you set up a condition in which the basal cells are killed, then these club cells here can actually go back to replacing many of the basal cells. They undergo a process of reprogramming back to forming basal stem cells. And we and others have shown that if we injure this alveolar region by treating the mice with a chemotherapy agent called bleomycin, which causes damage to this alveolar region, then club cells in these small airways can actually change their fate. They start to proliferate, and they give rise to type 2 cells. So this is a little bit different as well, rather unexpected. And again, we would like to try and understand the molecular basis of this. And then just finally, there's still a bit of an unknown, but mice that are treated with H1N1 type of influenza virus, there is damage to this region of the lung, and basal cells appear here in little pods, shown by Frank McCune, and nobody is quite sure where these basal cells come from. It may be likely that these club cells can go back to forming basal cells in this region. So there's a lot of interest here, and that's because if we could understand this kind of reprogramming, we might be able to promote it and to effect a better and quicker kind of repair. We also really want to understand what controls the proliferation and differentiation of these basal cells, because there are conditions which we can mimic in the mice by getting them to breathe sulfur dioxide, a low concentration of sulfur dioxide, mimicking conditions in human in which there's inhalation of toxic substances. And then these luminal cells are killed, leaving behind basal cells. And they leap into action, they start proliferating, and interestingly, they start spreading and quickly restoring a barrier function. Part of the function of this epithelium is to keep bacteria and nasty things out from getting into the underlying stroma and blood vessels. And so what these basal cells will do is that they change their shape and start to make connections between each other that they didn't have before to make a barrier. And then they make undifferentiated progenitors, and after a time they will start to differentiate into ciliated and secretory cells. And one of the interesting things is how they will make the right proportion of these cells. And again, we have evidence that notch signaling is involved in that process. So the epithelium does all this, and Jacques Gao, a postdoc in my lab, has been looking at a role of a transcription factor, a grainy head-like protein, in controlling the shape changes and adhesion of these cells. But there are other changes which are taking place in the underlying stroma. there's an influx of immune cells which we think are promoting the repair of this epithelium and are producing cytokines which to speed up this process. And there are also changes in stromal cells and in the blood vessels. And so in collaboration with Dee Gunn's lab at Duke, who had been exposing mice to more toxic gas, which kills both the luminal cells and disables some of the basal cells, it was found that the basal cells, if they can't proliferate, what happens is that there's an overgrowth of this energized stroma, and it ends up by causing a sort of fibrosis that will block these airways. And we think that this is what happens when the basal cells are not signaling and controlling the behavior of the stromal cells, and might mimic some conditions that happen in a condition known as bronchiolitis obliterans in humans after lung transplant, when probably some of these stem cells are being not as effective as they could be, leading to fibrosis. So we've wanted to understand what controls the proliferation and differentiation of these basal cells. So Jason Rock, a postdoc in the lab, set up this assay system, which we isn't high throughput, but it's kind of medium throughput, high content, as it's called, to screen for chemicals and growth factors that would stimulate the self-renewal of the basal cells. And we call this our tracheosphere assay. So what he did is he took advantage of the fact that the basal cells express a nerve growth factor receptor on their surface. We don't really understand why, but it's a very convenient handle for us to stain these dissociated cells with antibody and then use a process known as fluorescent activated cell sorting to pull out these basal cells, a pretty pure population. And then he dispersed these into these inserts, which contained that gel-like substance that Molly had used, in which we know that lung cells will grow if they're given the right conditions. And under these conditions, the factors we added, we can get about 10% of these basal cells to grow. And they grow and they form these little things which are now called organoids, or tracheospheres, and you can actually make a movie and watch the cells developing. From a single cell, we get a little cluster, and that cluster makes a little central lumen into which they're pumping fluid. And then if we take a section of one of these spheres, we see we've got basal cells on the outside. So this basal cell made more basal cells, but they also made luminal cells here. And by about 2 weeks, these luminal cells have differentiated into ciliated cells, marked here in purple. And you can actually see the little cilia beating. And they're also secretory cells marked in purple. The ciliated cells are purple. The secretory cells are making this glycoprotein called plunk, which they're secreting into the inside. So this is sort of further evidence that a basal cell is a stem cell that can self-renew and give rise to differentiated cells. Now, I told you to sort of remember about that FOXJ1 GFP mouse, because that's been very useful for assaying the production of ciliated cells. Because when the ciliated, although the basal cells are not green, because they're not expressing FOXJ1, by the time they have differentiated into ciliated cells, they're green. So Tomomi Tadakoro in the lab has used that to be able to screen for factors that will promote production of ciliated cells. So by day 12 in the control, rather few of these spheres have ciliated cells. But she found that when she added the cytokine interleukin-6, she could get premature and more abundant production of ciliated cells. And if she used an agent that blocks interleukin-6 signaling, then there were fewer ciliated cells, whereas the control by day 14 had lots of ciliated cells. And so then going back to in vivo, she asked what happens about IL-6 during this repair process, and showed that there was a peak of IL-6 production after about a day in the underlying stroma. And quite surprisingly, this interleukin-6, we thought it was going to be made by the immune cells. It turns out instead it's made by a subclass of the stromal cells. So this is important because it's telling us that some of these cytokines that had been shown before to be expressed in a wound repair situation actually have the capacity to work directly on the stem cells and to change their behavior and to promote the production of cilia, which might be useful early on to remove any nasty compounds. And so it might not be such a good thing to start knocking out all of these cytokines, which you think might be bad. You might want to keep some of them going in order to affect repair. Now, I probably have to hurry up here, but I just wanted to point out that there are other changes in this stromal population. And if any of you are interested in the aging of the lung, I'll refer you to this recent paper from my lab, in which we show very interesting, McCarron Wensley even showed, very interesting changes in the number of basal cells in the aging airways of the mice. And quite a remarkable change in the architecture of the epithelium. So instead of being all a nice layer like this, they were forming these strange little cysts that were underneath the epithelium. And she showed that this old stroma actually had a lot of lymphoid cells in there, even though these mice had been kept in the very, very best conditions, and as far as we know, were not being exposed to any known mouse viruses or flu or so on. So we really don't understand this yet, but we think that there are age-related changes in this epithelium that might make the epithelium leaky. And therefore, this stroma gets activated in some way. But I just wanted to point this out if some of you are interested in aging and also have studying old mice. Okay, so I'm just going to now talk about the very last few slides about studies we've been doing on the stem cell population in this alveolar region of the lung, which has a very different structure, these honeycombed-like areas in which there are type 2 epithelial cells, which make surfactant protein, and these very thin type 1 cells here. And so very little has been known about these behavior of these type 2 cells other than the fact that they make surfactant protein. But there were some indications that they could give rise to the type 1 cells slowly over time. But whether they could do this more, they could proliferate more actively, and whether they actually would migrate or move around the lung was not known. And that was in part because many of the damages that are done to the lung result in changes in the architecture in which it's quite difficult to follow the behavior of individual type 2 cells and ask whether they're able to self-renew and move around and to repair the damaged lung. So we set out to look at the behavior of these type 2 cells. I'm not going to go into this in detail, but essentially we made it so that we could label these type 2 cells with a reporter protein called TD tomato or tomato, depending on which side of the pond you are. But this is just showing that after giving a pulse of the drug tamoxifen to the mice, we can label about 85% of these cells which are making surfactant. We could label them with a tomato, and then they would continue to express tomato, whatever they did. Now, the other trick that Christina Barkaukas used, she's a physician scientist in the lab, a pulmonary physician, she wanted to try and challenge these type 2 cells to start proliferating and behaving as stem cells without damaging the architecture of the lung too much. So she hooked up this allele we had so that it would do two things. It would both lineage label or label the cells with the tomato, a lineage trace, but also using a genetic trick. We were able to sort of like surgically kill about half of these type 2 cells very cleanly so that they were eaten up by the macrophages and then disappeared. but they were leaving behind little empty niches, which I will explain a little bit more in the last few slides I'm going to show. So here we have a situation in which many of the type 2 cells are being killed, but there's a few remaining cells which are lineage traced with this tomato. So now, with using high confocal, resolution confocal microscopy, she could go in and say what happened to these individual type 2 cells. And she found, as I think we sort of expected, that early on there were little clusters of these type 2 cells, maybe extending around one alveolus. But we had quite a surprise when we looked at, after 3 weeks, because these individual clones, and we knew they were individual clones from other experiments we did with a confetti allele, but now this is an outline of the alveolus. They had spread quite extensively, and this is actually, we can just look at this in 3 dimensions very quickly. They had obviously spread much further away than we thought. And so what we think is that these cells, when the niches are empty, they can start proliferating and then migrate from one alveolus to another through little pores, which are known to be there inside, between the alveoli. So then we are sort of beginning to ask, where are these cells going and what are the niche that they are trying to find? And so the last couple of slides is going to tell you about experiments we've been doing to identify the cells which are close up to these type 2 cells and are maintaining them throughout the life of the lung, and are perhaps protecting them and feeding them lipid precursors. And it was described, I had a reference earlier, by classical anatomists, that was a population of lipofibroblasts that had moved into the lung during that process of alveologenesis that I described to you, and have taken up residence near these type 2 cells. They also have long extensions that touch the endothelial capillaries, touch the type 1 cells, and touch the pericytes. So this is a section of a lung that's been shown where these cells reside. And we were very fortunate to find that these cells express a PDGF receptor, a receptor for a growth factor known as PDGF receptor. And Phil Soriano had made a mouse in which they have got green fluorescent protein in their nuclei. So here would be one of these cells with a green fluorescent protein expressing in the nucleus. And you can see that they are next to cells which are expressing surfactant protein C, one here, and here, and here, and here. And they also make lipid droplets. So this suggested that these were a kind of niche cell for the type 2 cells. And so what Christina did was to set up an assay to ask whether individual type 2 cells could form an organoid equivalent to these tracheospheres that we'd had from the airways. And so she sorted out the type 2 cells, lineage trace with tomato, and sorted out these lipofibroblasts with their GFP, and mix them together in this gel-like substance in a little insert. And she found that there were these little alveolar spheres made. And in the almost last slide, this is just showing one of these alveolar spheres. Now they can see that they have a very different organization to the tracheospheres. There are more type 2 cells, and they are around the outside. But on the inside, there are delicate cells which have markers, this HOPX, for the type 1 cells. So they have, under these conditions, differentiated into type 1 cells. And so now we're using this assay, and this is the conclusion here, that we're trying to use this assay, again, to screen for factors like interleukins and growth factors that will promote the growth of these type 2 cells. We're asking whether we can put them back into the lung, whether they would reintegrate into a damaged lung. Because other people have suggested that endothelial cells are also important for the growth of the type 2 cells, Mei Chung is asking whether endothelial cells and these fibroblasts work together. And then finally, we're asking whether the type 2 cells, like the basal cells in the airways, regulate the behavior of the stromal cells. Because there are conditions in the human, a very nasty condition called idiopathic pulmonary fibrosis, in which there's overgrowth of stromal cells and making a lot of extracellular matrix. And there is reason to think that this might, under certain circumstances, be because the type 2 cells can't keep up renewing the epithelium well enough. So these are the kinds of experiments we're doing, hopefully to try and keep us breathing and blowing flowers through the spring air as long as we are able to. And I will finish here by, I think I've acknowledged everybody as I went along. This is the current members of my lab, and these are my past members, and these are people with whom we have collaborated. And of course, it's extremely important to recognize the people who have been funding this work. And in particular, I want to just point out that there is the NHLBI has formed a consortium, the Lung Repair and Regeneration Consortium, that has a website here that has got information on it if people are interested in some of the ongoing projects in other labs in the United States. And I think we're hoping to link up with people in Europe as well to look at the role of stem cells and other cells in the lung in repair. And also I get funding from the Ellison Medical Foundation for studies on the aging of the lung. So thank you very much indeed.
Other questions? Could I ask, in the flu-infected
Okay, yes, I knew you were going to ask about that.
Do all the cells in the alveoli get infected? I mean, is that...
Actually, it's the airway and the alveoli, both of which get infected. I'm not entirely sure which ones are killed, but in the mice, there is a mouse version of the H1N1, and there is quite extensive damage and inflammation, and then you definitely get these little pods forming. Sure. But I don't know what cells are killed preferentially. Okay. I know you can get these little pods also after bleomycin treatment. Christina has seen that as well. So it requires a very extensive damage, and inflammation probably is playing an important role.
And when you introduce these class 2 cells later, in the later experiments,
yes,
do they finish up also in the little sacs in the alveoli?
Well, we haven't. That's a proposed experiment to put them back into the lung. We haven't actually done that experiment yet, but we think it might be possible because Hans Cleaver's lab, or these people who've come from his lab, who've grown these little organoids from stem cells in the intestine, on the colon, they're actually able, what they do is they give mice anemonas of EDTA and then put these little organoids back in and then clog up the anus, and then they will actually implant and grow in the epithelium. So we think that it might be possible to put these things back into the lung. But the problem with a lot of people ask about cell therapy of the lung and attempts to make sort of lungs for transplantation. But I think you can see from this three-dimensional architecture, it's going to be very difficult to make sure that all of these airways are properly seeded, and in particular to get these blood vessels, these little fine capillaries. I think that's one of the stumbling blocks of recellularizing scaffolds, is to get the endothelial cells back into these capillaries so that they would function and get blood to the alveoli.
Am I missing someone?
Well, I think everybody is.
The fourth row back and the gentleman. No. Oh, sorry.
Okay, Vivian. Vivian's got a question.
How similar or different is the human lung system to the mouse?
Okay, well, that's an important question. And I did try and schematize that. I do have a slide somewhere which is showing the relative size of a mouse lung to a human lung, very different. But we think that the large airways of the mouse lung, which are about 1.5 millimeters in diameter, is about the same diameter as the small airways in the human lung. And there are basal cells, as I told you, in the mouse trachea. There are basal cells in the human small airways, and they express P63 and keratin 5, and you can isolate the basal cells from the human lung. You can grow them into these spheres. You can grow them in air-liquid interface. They will differentiate it in ciliated and secretory cells. And actually, Xiao with Scott Randell, who provides us with the human basal cells, has shown that grainy head is very important in that differentiation of the human basal cells. So at the cellular level, there's a lot of similarities, which mean we can translate findings with the mouse to the human.
Hello, I was wondering how the lungs are able to keep up with the damage caused by smoking.
All right, smoking, well, that's really bad. And actually, it probably, people have shown that after smoking, there are slow changes in the epithelium. It's not quite as dramatic as, say, when they're inhaling sulfur dioxide, and we get sloughing off of the total sheets of these luminal cells. It's probably a slower loss of the epithelial cells, and then maybe a slower replacement from basal cells. Ron Crystal in New York has looked at changes in basal cells in mice which have been, or in humans, sorry, he's gone into human lungs and with little brushings can take out the basal cells from the airways of the human lung and look at their gene expression patterns, and has shown changes in those gene expression patterns in smokers versus non-smokers. He's published that work. So there are changes in the basal cells, and presumably also eventually they become transformed and give rise to cancer.
Yes. You didn't want the question. No. Sorry.
Okay, good for you.
I think you said there were fibroblasts that came in to effectively repair or help in the repair of the type 2 cells. Are they there beforehand, or do they come in after damage? And if so, what signals them to come?
So that's an interesting question. I think I showed, in this process of alveologenesis, then the precursors of these fibroblasts come in because they express PDGF receptor alpha. And people have shown that mutant mice that lack PDGF receptor alpha lack some of these fibroblast-like cells in the lung. So they come in during embryonic development. The interesting question is whether they'll start to proliferate after damage. And we are just doing those experiments. We have started to lineage trace those cells, and then Christina is damaging the lung and saying, do they start to proliferate, and are they the cells that give rise to fibrosis in the lung? We think they are. Some work we've already published is that we think that they do get out of control under some circumstances and then start to make too much extracellular matrix and are bad players. So they can be good players, but they can be bad players as well. So they've got to be kept under control, like everything else that can be good or bad.
Yes, I have a question from me and Liz Robertson, in fact. We were whispering about this, and I hope it's a simple question. So I'm going to be running a marathon next year. I'm training already, and I'm hoping that my lung capacity will increase. What's the mechanism by which it will increase? Do you know?
That's a very important question, because we've actually written a protocol for running our mice on wheels. because we've decided that we're studying couch potatoes. I mean, and one of the, I keep on saying, you know, there's slow turnover in the lung. It's very difficult to see if type 2 cells make more type 1 cells unless you injure them and so on. But we would want to put them on wheels to see now whether there is sort of micro damage and whether we can get more proliferation. But it's known, one of the systems for looking at changes in the stem cell behavior in mice is to go in, take out one of the lobes, take out the left lobe, and then the right lobes actually increase in size, and they make more alveoli. They don't bud them off, but what they do is they make more of these little septi to now subdivide the bigger alveoli into more units. So you end up by having the same number of units in 2/3 of the lung. And people are very interested to know the type 2 cells do proliferate. They make more type 1 cells, we've found, and they more blood vessels grow in, and so on. So it's possible that if you work really hard, Jim, you might get, but the problem is you're constrained by the size of your thorax, where in that experiment, so you've just got to do exercises to get a bigger, you know, thorax as well.
Brigid, we're going to leave it there.
Okay.
Thank you very much for simplifying what is obviously a very complex system. And I'm sure we'd all like to thank you again. Thanks very much. So I think you're supposed to get the thing, but they haven't left it here. So before we go, I have the pleasant duty of handing over a certificate of some sort.
Thank you so much.
And a medal, which go along with this important lecture. Thank you very much.
Thank you very much. I'm the chair.
And that's,